There is a sealed wooden crate in the storage facility of the University of Pennsylvania Museum of Archaeology and Anthropology that has not been opened by an outside researcher in over 15 years. It is cataloged as PUM-4-1974-9. Inside the crate is a skeleton. The skeleton was excavated from a royal tomb at Tel al-Lahm in southern Iraq in 1974 by a joint American-Iraqi expedition. The bones are well preserved. The tomb was undisturbed when the excavation team broke through the ceiling wall. Most of what was found inside has been displayed, cataloged, or published. The skeleton has not been displayed. The skeleton has not been published. The official reason on the file card is that PUM-4-1974-9 is awaiting further conservation analysis. That note has been on the card for 15 years. It is also not true. If you want to see more deep dives into archaeological discoveries that mainstream institutions would prefer you forget, hit subscribe. I cover this kind of content every week, and what's in this video is going to change how you think about ancient royal burials. The skeleton belonged to a man identified by the cylinder seal in his hand as Lugal-Anbara, a king of a small but wealthy city-state in the southern Tigris floodplain during the early dynastic 3A period. Carbon dating places his death at approximately 2600 BCE. He was about 55 years old when he died, taller than the average Sumerian male of his era, and in robust health for a man of his age. There is no obvious cause of death visible in the bones. The tomb itself was unusual in ways that have never been satisfactorily explained. Most early dynastic royal tombs follow a predictable layout: a primary burial chamber, an outer antechamber, and a single sealed entrance. The Tel al-Lahm tomb had two sealed chambers behind the primary one, both empty when the excavation team reached them, and both inscribed on their interior walls with cuneiform that translated as "for the second" and "for the third." The chambers were identical in dimensions to the primary burial chamber. Each contained a stone plinth of the kind ordinarily used to support a sarcophagus. Neither plinth showed evidence of ever having held one. The 1974 field report noted the inscriptions in the empty plinths and moved on. The lead archaeologist, Dr. Robert Hammond of Penn, recorded a single line in his personal notebook regarding the chambers. 3 rooms, 1 body, question for later. Hammond died in 1989 without returning to the question. No researcher since has revisited it either. Lugal-Anbara was buried with the standard markers of high-status interment for the period: gold ornaments, ceremonial weapons, lapis lazuli inlays, and the remains of 6 attendants who appear to have been ritually killed and placed alongside him. For most of the skeleton, the analysis was unremarkable. Sumerian royal burials of this period have been studied for over a century. Lugal-Anbara fit the pattern. The unremarkable assessment ended in 2008. That year, a paleoradiologist named Dr. Maya Levin, working out of a temporary research arrangement with the Penn Museum, was given access to PUM-4-1974-9 as part of a broader project digitizing Bronze Age skeletal collections using high-resolution computed tomography. The project was meant to be routine. The CT scanner was a new acquisition. The museum wanted as many of its cataloged specimens scanned as possible before the visiting equipment was returned to the manufacturer. Levin scanned the Lugal-Anbara skeleton over 3 sessions across 2 weeks in February 2008. She paid particular attention to the rib cage because Bronze Age cardiac archaeology was a niche specialty of hers, and she had been hoping for a usable thoracic specimen. What she found was not usable for the project she was working on. It was not usable for any project she had ever worked on. The CT scans of the chest cavity showed 3 distinct anatomical structures consistent with the morphology of cardiac tissue. The structures were spaced unevenly along the anterior thoracic axis. Each was associated with its own arterial branching pattern, visible as calcium deposit traces in the surrounding tissue matrix. The arrangement was not random, it was structured. It looked to Levin like a body that had been built to support 3 separate hearts. She did not believe this. She rescanned the specimen twice. The structures persisted. She brought in a second radiologist from Temple University to verify. He confirmed the imaging. He could not explain it. She then sent anonymized scan data to a colleague at Johns Hopkins who specialized in pediatric cardiac anomalies, asking only whether the configuration was consistent with any known developmental pathway. The colleague's reply, which Levin has since shared with at least 2 independent researchers, said the configuration was not consistent with any pathway he had ever seen, and that he would be very interested in seeing the original specimen. Levin then sent the same anonymized data to a forensic radiologist in Zurich who had no prior involvement with Bronze Age archaeology, and who therefore had no professional reason to assume the imaging belonged to an ancient subject. The Zurich response asked whether the specimen had survived its own surgery, because no living patient with that thoracic configuration could have grown to maturity without medical intervention that did not exist in 2008, let alone 2600 BCE. Levin filed an internal report at the Penn Museum in March 2008, describing her findings and requesting permission to publish. She did not receive permission to publish. What she received instead was a meeting with a senior curator and a representative from the museum's Research Ethics Committee. The meeting lasted approximately 90 minutes. At the end of the meeting, Levin was informed that the imaging artifacts she had identified were almost certainly the result of post-depositional mineralization, that her interpretation was scientifically irresponsible, and that the museum would be reclassifying the specimen as having non-standard preservation features requiring further analysis. The classification was finalized in June 2008. The skeleton was returned to its crate. The crate was returned to storage. Levin's CT scans were retained in the museum archive but not made publicly accessible. Levin's research arrangement with Penn was not renewed. She has not spoken publicly about Lugal-Anbara since 2010. To understand what the museum's reclassification was actually doing, you need to understand what it was rejecting. Post-depositional mineralization is a real phenomenon. When organic material decomposes inside a sealed burial environment, dissolved minerals from the surrounding soil can precipitate inside the cavities left behind. The result on a CT scan is a faint trace of the original soft tissue structure preserved as a mineral cast. It is the standard explanation for unexpected internal features in well-preserved skeletal specimens. It is also, in this case, the wrong explanation. Mineralization casts are diffuse. They follow the contours of whatever cavity contained the original tissue. They do not produce 3 discrete, structured anatomical formations with independent arterial signatures. The pattern Levin documented was not a mineralization cast. It was an architectural feature of the body itself, preserved by the unusually dry conditions of the Tel al-Lahm tomb. The skeleton was telling her something about how the man had lived. The reclassification refused to listen. Now we need to talk about what 3 hearts in one body would actually mean. In standard human development, the heart forms from a single embryonic structure called the cardiac tube, which folds and septates into the 4-chambered organ we are familiar with. Anomalies of this process are well documented. Cor triatriatum, in which a single chamber is divided by an internal membrane to form a 5th functional cavity, occurs in roughly 1 in 100,000 births. Heterotaxy, in which cardiac structures are mirrored or duplicated, is rarer but documented. None of these conditions produce 3 discrete hearts. None of them produce 3 sets of independent arterial trees. There is one biological mechanism that theoretically can. It is called tetragametic chimerism. Chimerism occurs when 2 separate fertilized eggs fuse during very early embryonic development into a single organism. The resulting body contains 2 complete and distinct genomes, intermingled at the cellular level. In the rare cases where chimerism affects cardiac development, the embryos can fail to fully merge their cardiac structures, producing duplicate or partially independent cardiac tissue. Tetragametic chimerism is real. It is documented. The case of Karen Keegan, identified in 2002 when DNA tests during a kidney transplant evaluation indicated she could not be the biological mother of her own children, established the condition in modern medical literature. The case of Lydia Fairchild in 2003, who faced welfare fraud charges for the same reason, brought it to public attention. Since then, several dozen subsequent cases have established that some living humans walk around with two complete genomes in different parts of their bodies. The condition is undetectable without specific genetic testing, and the medical community now suspects it is significantly more common than once believed. A 2015 review in the journal Human Genetics estimated that as many as 1 in 8 individuals from naturally conceived pregnancies may carry minor chimeric tissue, with full tetragametic chimerism likely being orders of magnitude more common than the handful of recognized cases would suggest. In 2017, the American singer Taylor Mull publicly revealed that DNA testing had confirmed she carried 2 complete sets of DNA, the result of having absorbed her unborn twin during early embryonic development. Mull's body shows the chimerism visibly. Her torso is divided down the center line by a clear pigmentation difference, with her left side carrying her sister's genetic profile and her right side carrying her own. Cases like Mull's have moved chimerism from the category of medical curiosity into the category of recognized human variation. Three Hearts, however, is not on the documented chimerism spectrum. 2 embryos producing 3 cardiac structures would require an additional anomaly during the merging process, or a different mechanism entirely. There is no living human on record with 3 functional hearts. There is no known biological pathway that produces them. There is a Sumerian king in a crate in Philadelphia who appears to have had 3. Now we need to talk about the genetics. In 2011, 3 years after Levin's CT findings were reclassified, a graduate student in the Penn Anthropology Department named Daniel Ortiz submitted a proposal to perform ancient DNA analysis on a small sample taken from the molars of PUM for 1974-9. The proposal was approved through normal channels. Ortiz was not aware of the Levin episode. The skeleton's reclassification meant that newer researchers did not necessarily encounter the earlier history of the specimen. Ortiz extracted DNA from 2 separate molars over the course of 2012 and submitted the samples for sequencing at the Max Planck Institute for Evolutionary Anthropology in Leipzig, which had recently established itself as the world's leading center for ancient DNA work. The samples were processed alongside a batch of other Bronze Age Mesopotamian specimens. The results came back in early 2013. The 2 molar samples produced different genomes, not subtly different, not within population different, significantly different at the level of distinct ancestral lineages. One sample showed standard Bronze Age Mesopotamian ancestry, with the expected mixture of Levantine farmer, Iranian highland, and Caucasian hunter-gatherer components. The other sample showed a base layer of similar ancestry, but overlaid with a substantial genetic contribution from a population that the Max Planck pipeline could not match against any reference genome in its database. The unmatched component was not Neanderthal. It was not Denisovan. It did not match any of the known ghost lineages that have been inferred from anomalies in modern African and Eurasian DNA. It was something else. The signature accounted for nearly 14% of the genome in the affected molar, which is far above any plausible level of contamination. The component was structured, meaning the alleles within it showed internal correlations consistent with a real ancestral population rather than the random scatter that contamination produces. Ortiz was excited. He believed he had stumbled into something significant. He began drafting a paper. He did not finish drafting the paper. In late 2013, the Penn Museum issued a formal notification stating that the original DNA samples from PUM-4-1974-9 had been compromised by laboratory contamination, that the results were therefore unreliable, and that no further sampling of the specimen would be authorized. Ortiz was reassigned to a different project for the remainder of his graduate work. The Max Planck reports were not formally retracted because they had never been formally published, but copies of the raw sequence data were quietly removed from the institution's open access archive. Ortiz still has his personal copies. He has not made them public. He has, however, shared them with a small number of independent researchers, and the unmatched genetic signature has been re-examined by at least 3 teams. None of those teams has been able to identify it. None of them has been able to refute it either. In 2019, a forensic geneticist at the University of California, Berkeley named Dr. Helena Vargas, who had received the Ortiz dataset through a colleague, submitted a request to the Penn Museum to perform fresh sampling on PUM-4-1974-9 using more advanced methods that had become available in the years since the original work. Her stated goal was to either confirm or refute the unmatched signature using a clean extraction protocol. Her request was acknowledged. It was reviewed. It was eventually declined on the grounds that the specimen was undergoing long-term conservation work that precluded destructive sampling. Vargas received the decision letter in early 2020 and has not pursued the matter further publicly. The conservation work the letter referred to has not been publicly described, and the specimen has, by every available account, remained in its sealed crate throughout. The signature is real. It belongs to no known human population. It was found in the body of a Sumerian king who appeared on imaging to have had 3 hearts, and every researcher who has tried to investigate it further has been redirected, declined, or quietly defunded. There is a context for this that bears stating plainly. The Sumerian king lists the genealogical records that the Sumerians themselves maintained divide the rulers of their early dynasties into 2 categories. There are the kings who ruled before the flood, who reigned for tens of thousands of years, and whose names are accompanied by descriptors that translate as the Shining Ones or the Seed of the Heavens. And there are the kings who ruled after the flood, who lived ordinary human lifespans and whose ancestry is described in conventional terms. Lugal-Anbara, by his dating, sits in the after the flood category, but his tomb seal includes an unusual epithet of the older blood. The phrase appears occasionally in early dynastic texts. It is generally interpreted as a metaphorical reference to ancient lineage or noble descent. It has not been interpreted in the academic literature as a literal claim about biological inheritance. There are reasons to reconsider that interpretation. A handful of Sumerian medical and ritual texts describe royal anatomy in terms that do not match the anatomy of ordinary subjects. A tablet from Shuruppak, dated to roughly 2500 BCE, describes the king's body as containing doubled organs that the common man does not bear. A ritual text from Lagash refers to a class of high officials as those whose chest holds more than the chest should hold. A funerary hymn for an unnamed early dynastic ruler, recovered from Nippur and currently held at the Istanbul Archaeology Museums, addresses the king directly with the line, "3 lights you carried within you, 3 lights returned to the heavens at your dying." The standard scholarly translation renders lights as a metaphor for spiritual essence. The Sumerian word being translated, however, also appears in medical texts where it unambiguously denotes a vital internal organ. These passages have traditionally been read as poetic flattery or religious symbolism. They could also be read as literal description. The literal reading was not seriously considered, while the only available evidence was the texts themselves. The literal reading became harder to dismiss in 2008 and harder still in 2013. The CT scans, the genetic results, and the seal's unusual descriptor do not, on their own, prove anything. But they sit together in a way that is difficult to dismiss. A man with anomalous cardiac anatomy. A genome that includes contributions from a population modern science cannot identify. A self-description that claims older blood than his contemporaries. Texts from his civilization describing royal bodies that contain doubled organs. And a museum that has reclassified, sealed, or quietly buried every line of evidence that pointed toward a coherent interpretation of all 5. Lugal-Anbara is not the only Sumerian royal skeleton with anomalies of this kind. A 1969 excavation at Ur, conducted by a team from the Iraqi Department of Antiquities, produced a skeleton with a fully duplicate set of inner ear bones, packaged in a structure unlike any documented in modern human anatomy. The duplicate auditory ossicles were not loose fragments. They were articulated within an additional bony housing carved into the temporal region of the skull, in a position that suggested they had been functional during the subject's lifetime. The bones were originally interpreted as an artifact of mishandling during recovery, on the assumption that fragments of a second specimen had been mixed in with the first. No second specimen was ever identified. The skull remained intact. The duplicate bones remain in storage at the Iraq National Museum, last examined in 1971 by Dr. Faisal al-Wakil, whose final report concluded only that the configuration cannot be explained by current anatomical models before being filed and forgotten. A 1977 burial at Lagash, excavated by a French team, produced a king whose dentition included a third molar row behind the standard two. The condition is currently classified as a developmental abnormality. It has not been reanalyzed using modern imaging techniques. The skeleton is held at the Louvre. A 1991 emergency salvage dig in Babylon, conducted in the chaos preceding the First Gulf War, recovered a body with what the field report described as supernumerary major joints in the lower limbs. The phrase was never elaborated in any subsequent documentation. The single page of original field notes that survived the war includes a sketch of one leg showing what appears to be an additional knee joint between the standard knee and the ankle. Whether the sketch represents the excavator's interpretation or a genuine anatomical observation cannot now be determined. The specimen was relocated to a storage facility outside Baghdad and has not been located by any researcher since the looting of the Iraq National Museum's storage annexes in April 2003. It is presumed lost or destroyed. Each of these specimens, like PUM-4-1974-9, has been given a conservative explanation and removed from active research circulation. The pattern is not chaos. It is a specific category of anomaly. Bodies that look human, that were buried as kings, and that contain structural features modern medicine has no normal pathway to produce. If even one of these reclassifications is wrong, the implications are uncomfortable. The Sumerians would not be the only ancient civilization to claim that their early rulers were of a different biological order than their subjects. They would be one of many. They would also be the first whose claim could be tested with modern imaging and modern genetic analysis. And the testing, in at least one well-documented case, came back with a result that the institution doing the testing chose to bury rather than publish. Lugal-Anbara is in his crate. The CT scans are in the archive. The Leipzig sequence data is on Daniel Ortiz's personal hard drive. Helena Vargas's 2020 decline letter is filed in a Berkeley office. The cylinder seal that names him of the older blood is on display in the museum's main Mesopotamian gallery, where most visitors do not know what those words refer to, and most curators are not asked. The reclassification is the most important part of this story, not because it conceals an unambiguous truth, but because it forecloses an investigation that should have continued. The museum did not say, in 2008, that Levine was certainly wrong. It said her interpretation was irresponsible. It did not say, in 2013, that Ortiz's results were definitely contaminated. It said the samples were unreliable. It did not say, in 2020, that Vargas would never receive sampling permission. It said the specimen was in conservation. Each response was procedurally appropriate. Each response also made the question disappear. We do not know whether Lugal-Anbara had 3 hearts. We do not know what the unmatched genetic component in his DNA represents. We do not know whether his older blood was a metaphor or a genealogical fact. We know that the questions exist. We know that the evidence in raw form is on tape and in storage. We know that the institutions in possession of that evidence have, repeatedly, chosen reclassification over investigation. Whether that choice was scientific caution or something else is a question each viewer is entitled to answer for themselves. The body is in Philadelphia, the samples are in Leipzig, the seal is on display 300 feet from the crate that holds the man it names. None of those things will change without external pressure. Until they do, Lugal-Anbara remains exactly where the museum has placed him. In storage, off the record, and quietly waiting for someone to ask the questions everyone before them has been redirected away from. If you want to see more ancient mysteries that have been quietly removed from the public record, subscribe.